Hey there folks, and welcome to the second video for Chapter 5, The Working Cell. This video is going to wrap up our work with Chapter 5. Now this video is a little bit more technical. Hang in there with me, because before we can keep discussing how our cells use energy and bring things into the cell in order to use them for energy, we actually need to talk about energy itself for a moment, and that means backing up and discussing physics for a second. There are two major types of energy, kinetic and potential. Kinetic energy, energy first, is the ability to have an effect on other things, the ability to get work done. Kinetic energy is the energy of things that are moving. An example of this is thermal energy, which is the energy held by atoms and molecules, and it's generated by their random movements as they vibrate. Potential energy is the energy that something has either because of its location or because of its structure. An example of that structure, potential energy, is again in molecules. When our cells are undergoing cellular respiration, they break down glucose and use the energy that was in those bonds to help generate ATP. Heck, on the flip side of this, when our cells are using practically any process that requires a push, they'll bring in enzymes or ATP, and by breaking the bonds of ATP, they release energy, and it's the potential chemical energy of that phosphate bond in ATP that contributes energy to another process so it can continue. When energy is contributed from one thing to another in this fashion, some of the energy that gets contributed is lost in the form of heat. And the movement of this energy, this heat energy, and where it goes and how constant it is, plays into energy transformations, and specifically in thermodynamics. That's what thermodynamic means. It's the movement of heat. When we are talking about the play of energy in our cells, something to consider are the first two laws of thermodynamics. The first law says that all energy in the universe is actually constant. It never disappears. It changes form from potential to kinetic. Say when you start a car rolling downhill, from potential because of its location to kinetic when it starts moving. And it may transfer from one place to another. In the case of cellular respiration, it may move from ATP into part of the Krebs cycle. Keep that running. However, it never actually disappears. When things do move, when energy moves, it can, however, increase the energy of a system, and this can increase the chaos or the randomness associated with the system, at which point we are subject to the second law of thermodynamics, which says, yes, all energy in the universe is constant. However, it is constantly moving toward a higher entropy, and entropy is a term for randomness or chaos. So then basically there is all this energy in the universe, and it is constant, but it changes shape, it changes type of energy, it changes location, and the more that it changes, the more chaotic things become. When our cells are using this energy, there are two major types of chemical reactions that they undergo: exergonic and endergonic. An exergonic reaction is one where the reactants at the beginning of the reaction have more energy than the products do at the end. Or another way of looking at it is, through the course of the chemical reaction it's going to take these reactants and turn them into these products, we lose energy. Endergonic means that we gain energy when we make products from certain reactants. All right. Exergonic reactions tend to happen much more easily, and they tend to even be spontaneous. Endergonic, because you need more energy put into the reaction in order to get to the products, are not spontaneous reactions. They take energy and time. When molecules are broken down for their energy, in a biological setting, this is what metabolism is. It's our bodies, our cells breaking down energy, breaking down bonds of molecules in order to get energy from them. And the technical definition of a cell's metabolism is the sum total of all exergonic and endergonic reactions that it undergoes. Likewise, for our whole bodies at a much larger level, it is the sum total of all exergonic and endergonic reactions that we undergo. A metabolic pathway is any series of chemical reactions that either build a complex molecule, say building complex proteins to build muscle out of, or break it down, like when we digest food and use the energy from that food to run our bodies. Energy coupling is the use of energy released from exergonic reactions to drive endergonic ones. That energy has to come from somewhere, right? First law of thermodynamics. Energy is constant in the universe. So the energy that goes into our endergonic reactions must come from somewhere. That isn't to say that all reactants require the same amount of energy in order to run their reaction, nor is it to say that the energy that's required to kickstart a chemical reaction can't be modified. Biological living things make enzymes, which are a type of biological catalyst. In terms of chemistry, a catalyst is a molecule which helps kickstart a chemical reaction by reducing the amount of energy that is required to get it going. That energy is called the activation energy. It's the amount of energy required to activate the cellular or chemical reaction. The difference between an enzyme and one of those cellular molecular battery molecules like ATP that we've talked about before is ATP is changed by its interaction with a reaction. It loses a phosphate group. But enzymes are a type of catalyst, and catalysts are not changed by interacting with a reaction. Here on the left, you can see the difference in a pretty simple reaction that having an enzyme makes. With an enzyme, the activation energy is sometimes much lower than the activation energy of the same reaction without an enzyme, and overall, the product still has the same amount of energy when you're done, but having and using enzymes in a reaction can save your body or your cells a significant amount of energy to be invested elsewhere. By the way, not all enzymes necessarily function the same way, but here are some of the big common factors. When an enzyme becomes active in a reaction, there are substrates, certain other molecules, that bind to places on the enzyme called an active site. You can see this happening here in the diagram to the left. When the substrates form the product of the reaction, they are then released from the enzyme. But you see that the enzyme itself never changes shape and it never breaks apart. There are specific conditions that are best for an enzyme, however. Enzymes can have induced fit, which means that specific enzymes may bend a little bit, change shape a little bit in order to lock substrates in place. So the enzyme may change a little bit so it can better fit the substrate. Some enzymes function best under particular temperature conditions or pH, like say the enzymes inside your body will function best at your body's average temperature and at the pH they're supposed to. And some enzymes themselves need a little help. These are called cofactors, inorganic cofactors, things that can be ions of certain types of metal, can bind to, say, the backside of an enzyme and cause induced fit or help the enzyme function. When those cofactors are themselves organic molecules, which means that they're made out of carbon instead of a metal or some other element, they are called coenzymes specifically. Our cells need to be able to control their enzymes, however, and this means that sometimes it's necessary to inhibit or turn off enzyme function. Say when a process, a reaction has run long enough and enough product has been made, it's time to turn off the machine that is the enzyme. And there's a couple of different ways of doing this: competitive and non-competitive inhibitors. Competitive inhibitors function by binding with the enzyme in place of the substrate. That's that pink diamond, pink triangle side of this diagram over to the left. See how the pink triangle marked I for inhibitor binds in the same place that the substrate is supposed to? That's the active site. And by binding in place, it competes with the substrate for being able to bind to enzymes. And that slows down how much of the substrate is made and eventually will turn off the process, or at least fine-tune it. Non-competitive inhibitors, like the right side of the diagram, don't bind to the active site. They bind somewhere else on the enzyme, but they change the fit of the active site so that the substrate can't bind there anymore. And that has the same effect of slowing down or turning off the process. Feedback inhibition is another way of using competitive and non-competitive inhibition. Feedback inhibition functions by triggering inhibitors when there's so much of the substrate floating around that inhibitors begin to be manufactured and either compete with the substrate or bind to the backside of the enzyme. And they will keep doing this until the substrate levels drop down to a point where the cell is directed to stop making the inhibitors, at which point those drop down and the substrate can build back up again. And they go back and forth, maintaining a sort of dynamic balance. Last but not least, we also use inhibitors, not just the ones that we make in our bodies, but we use them to help our bodies do certain things. Say in the form of medications. Things like penicillin, which is our very first antibiotic, is actually a type of enzyme inhibitor. So are things like ibuprofen, which is a common pain relief medication. However, as so frequently happens, something that can in small amounts be a medicine, in larger amounts or uncontrolled amounts can be very dangerous. And there are enzyme inhibitors in pesticides and in poisons that are designed to, say, keep insects or weeds from growing out of control. But if something else were to be exposed to these compounds, it would still inhibit their enzymes, sometimes with very dangerous results. All right, folks, thanks for hanging in there. I know this one was a little bit more technical. We'll talk about enzymes and inhibition. This wraps up Chapter 5. Nicely done. I will see you folks next time. Take care.